Hello, my name is Rashmi Bhat and I'm presenting a part of my PhD research on how landscape structure and biodiversity influence tick abundance in the Western Ghats of Karnataka in southern India. The Western Ghats is one of the world's 8 hottest biodiversity hotspots, but it is also a region where humans, livestock, and wildlife increasingly interact in fragmented landscapes. These interactions are particularly important in the context of the Kyasanur Forest Disease or KFD, a tick-borne zoonotic disease endemic to this region. However, we still have limited understanding of how ecological factors, especially wildlife communities and land use patterns, shape tick abundance and by extension disease risk. In order to understand a part of the puzzle, I conducted a landscape scale study across the KFD endemic districts using a gridded sampling design covering 63 grids. Within each grid, we deployed 3 camera traps to quantify wildlife species richness and relative abundance between January and May 2023. At the same time, we conducted tick flagging surveys along 600-meter transects to estimate tick abundance. In total, we collected over 65,000 ticks across the study area. These results show clear spatial patterns in tick abundance across districts and sub-districts. First, as can be seen in the right-hand side top figure, nymphal tick abundance varied significantly across forest categories, indicating that landscape structure plays an important role in shaping vector populations. Second, wildlife species richness was positively associated with nymphal tick abundance. Sites with higher wildlife richness tended to have higher tick abundance, with our models indicating approximately a 12% increase in expected nymphal abundance for each additional wildlife species detected, as seen in the right-hand side bottom figure. Third, as seen in the central figure, tick abundance peaks in wildlife-livestock interface areas with moderate domestic activity and high wildlife richness, rather than in purely wildlife or livestock dominated systems. In contrast, domestic animal activity did not show a significant association with tick abundance, nor did it vary substantially across spatial scales. This suggests that in this system, wildlife communities are more influential than livestock in driving tick dynamics. Overall, our findings highlight that forest structure and variation in wildlife communities are key drivers of tick abundance in KFD endemic landscapes. This has important implications for understanding spatial heterogeneity in disease risk and for designing targeted surveillance and intervention strategies. Thank you for your attention.